We've looked at the structures of fats and oils. We know they're esters. So now we're going to look at how unsaturation can affect properties such as the melting point. Fats such as butter or animal fats tend to be solids at room temperature. Oils such as olive oil and sunflower oil tend to be liquids at room temperature, as you can see. So why do fats have higher melting points than oils? To understand why edible fats have higher melting points than edible oils, we need to look at the structure of the fatty acid chains present in the fat molecule. And in particular, we're looking for whether there's carbon-to-carbon single bonds, i.e. is it saturated, or carbon-to-carbon double bonds, i.e. is the molecule unsaturated. So let's say we have a saturated molecule. You can see the molecule's fairly straight and linear, and this has implications for how the fats pack, because if the fatty acid chains are nice and straight, as you can see here, it means you can pack them quite closely together. Therefore, if there's lots of overlap and therefore there's lots of intermolecular forces between the fatty acid chains. Now let's look at the structure of edible oils. So oils tend to be unsaturated. They tend to have at least one double bond in the carbon chain for the fatty acid. So I'm going to stick a double bond in here. And this tends to cause a bend or a kink in the molecule. So if I introduce a bend into the molecule, you can see that I can't pack these fatty acid chains so close together. This means there's less overlap between the fatty acid molecules. Therefore, there are less intermolecular forces to break. So these will, the bonds, if you like, between the fatty acid molecules be weaker, and therefore will require less energy to overcome. So to summarize, you can see that the oils are much less compact than fat molecules and cannot pack together as closely as these fat molecules. So this implies that the intermolecular forces, mainly London dispersion forces between the oil molecules, are weaker and therefore easier to break than those of fat molecules, explaining why fats have higher melting points than edible oils. So we can say that the more single bonds between the carbons, i.e. the more saturated the fat, the higher the melting point. Or the more unsaturated the molecule, the lower the melting point. So we can assume that fats are more saturated than oils, or conversely, oils are more unsaturated than fats. The reason that saturated fats have higher melting points is due to the fact that the chains are straighter and you can pack them more closely. If you can pack them more closely, then there's more overlap and there's larger amounts of intermolecular forces between the fat molecules, typically London dispersion forces. The more unsaturated the molecule, in terms of fats and oils, therefore the more carbon-to-carbon double bonds you've got. If you've got the more carbon-to-carbon double bonds you have, then this is going to introduce bends and kinks into the molecule. This means that these molecules pack less closely together. That means you've got a smaller amount of intermolecular forces, such as London dispersion forces, between the molecules. This means that it will take less energy to separate the molecules, and therefore you're going to get lower melting points. Note, I'm not talking about breaking covalent bonds, I'm talking about separating molecules. So to summarize this part, edible oils have lower melting points than edible fats. The double bonds or unsaturation present in fatty acid chains prevent oil molecules from packing closely together. So the greater the number of double bonds you've got present, the weaker the London dispersion forces or van der Waals forces of attraction between the molecules. So to summarize, the greater the degree of unsaturation, i.e. the more double bonds you've got, the lower the melting point. The animation shown next will give you an idea of how this can affect melting point. So we've seen there is a relationship between the melting point of a fat and oil and the degree of unsaturation, i.e. the amount of double bonds present in the molecule. There is an experiment we can do to get an idea of the amount of double bonds present in the molecule, and what we can do is we can do the test we looked at National 5 and add bromine in an addition reaction. So the more bromine that can be accepted, the more double bonds present. So if I take something like lard, which is a high melting point, it's pretty saturated, hardly any double bonds, and it will accept about 1 drop of bromine that can be decolorized before the bromine color stays as you add more drops. If you take margarine, which might have 1 or 2 more double bonds present, it might be more unsaturated, then it might take 2 drops of bromine, as it might have more double bonds that can accept the bromine. And if you take something like olive or corn oil, which has a low melting point, that means that it has a high degree of unsaturation, i.e. lots of carbon-to-carbon double bonds. Therefore, it can decolorize more bromine than something like lard, which might be more saturated. So we can conclude from experimental data that unsaturated compounds, i.e. compounds that have lots of carbon-to-carbon double bonds, will quickly decolorize bromine solution. And this is because the bromine molecules take part in an addition reaction across the carbon-to-carbon double bond. We can say, therefore, that the greater the number of carbon-to-carbon double bonds present in a substance, the more bromine can be decolorized. So the amount of bromine that you can be decolorized can be used to get an idea of how many double bonds are present within a molecule. So let's compare 2 fatty acids. This fatty acid has got 1 double bond present in it. So we would call that a monounsaturated fatty acid. This means that it can accept 1 molecule of bromine in an addition reaction across this double bond in an addition reaction. So you can see that the double bond is broken and we've accepted 2 atoms of bromine across that double bond. If we have a polyunsaturated fatty acid, so we've got 1, 2, 3, 4 double bonds in here, then I can attach a bromine molecule across each of these double bonds. In total, I can have 4. So when the double bond breaks and we add the bromine in an addition reaction, we get this being formed. So you can see that a polyunsaturated fat or oil can accept 4 times, in this case, the amount of bromine molecules compared to one with just 1 double bond. So you can use the bromine addition reaction to get an idea of how many double bonds are present within the molecule. So here's a quick question for you to try. To which family of organic compounds do triglycerides belong? So you've got a triglyceride, this molecule here, a fat or an oil. Well, you can see that it's an oil, it's got 3 ester links, so we call this an ester. You wouldn't call it a fat or an oil. So a family of compounds are esters, not fats or oils. So here's a question from an SQA specimen paper. Pause the video, have a go at it. It's a 2 marker and see if you can get the correct answer. Okay, so why does the melting point of shea butter higher than that of edible oils at room temperature? The 2 points you're looking for is shea butter has more saturated, has fewer double bonds, it's not very unsaturated, 1 mark. And your second mark is talk about the London dispersion forces or Van der Waals forces between the molecules are stronger than in oils, therefore the melting point is higher. So those are the 2 points you need to be aware when you're describing the properties of fats and oils. This is the final learning outcome for fats and oils in terms of their role in the body. Fats and oils are a concentrated source of energy. Fats will yield more energy per gram than carbohydrates or proteins. So that's the main function of fat in the body in our diet. Another thing is they're a source of fat-soluble vitamins in the body. So we'll see an example in the next slide. So if you look at vitamin A is basically mostly hydrocarbon. Most of that molecule is non-polar. I know there's an OH at the end, but if you look at the majority of the volubule, this is generally a fat-soluble vitamin. So we need fats to dissolve our vitamins in. However, if you look at vitamin C has 4 hydroxyl groups, and this is a water-soluble vitamin. So this reinforces the bonding part we did earlier, that if you have lots of hydroxyl group, it's a polar molecule, it will dissolve in water. But if your molecule is mainly hydrocarbon, it is a non-polar molecule, and therefore it will tend to dissolve in fats and oils. So you can see that the vitamin A is fat soluble, mainly hydrocarbon groups, non-polar. However, vitamin C, which you often find in juices which are quite full of water, contains several hydroxyl groups which can hydrogen bond with water, so they're polar. Remember, like dissolves like. This is just going to relate to the bonding that we covered earlier. The thing to note about a fat molecule is that it has no hydroxyl groups on it. There's no OH groups. So that means that fats and oils don't dissolve in water. You know that oil and water don't mix. And the reason is because most of the molecule is non-polar. We're only going to get London dispersion forces between the molecules. So because they don't really have any polar hydroxyl groups or ions and cannot dissolve in water, which is a polar substance, we describe fats as being non-polar. So this is a final summary of the learning outcomes for fats and oils as part of the ester section. Have a look at them, make sure you're comfortable you know what these mean and understand them. Maybe have a quick look in bright red, and then when you're ready you can have a go at the quiz and homework exercises posted next.